All right, my name is Stephanie Hicks. I'm an assistant professor at Johns Hopkins, and I'm sort of introducing this next section of the summit. The title of my talk is Orchestrating Single Cell Analysis with Bioconductor, and a lot of the material that I'm going to cover here you'll see in future talks in a few minutes. But the reason it's called, the title of this is orchestrating, is because the mental image that Bioconductor is aiming for is that there are a lot of tools and packages for the analysis of genomics data, similar to many musicians in an orchestra. And to make them work together, Bioconductor really functions as a conductor of the orchestra, and it provides this flexible platform to integrate everything together. So in 2015, Wolfgang Huber and many others wrote this really great paper titled Orchestrating High Throughput Genomic Analysis with Bioconductor. And it was a really foundational paper for me when I was really starting to get into Bioconductor. It does a great job of describing what is Bioconductor, how users can get into it, and how developers can contribute to it. At the time, there were around 900 packages. Now there are over 1,700 packages. And then it also describes the different types of data, high throughput sequencing data that Bioconductor supported. And so this was really great at the time. And then around 2014, the world of single cell analysis really started to pick up. And I think developers in Bioconductor really saw that Bioconductor was a natural home for high throughput single cell software development. And I think they did this for two reasons. One, that Bioconductor was really a pioneer for the analysis of what I would refer to as low throughput single cell analysis data or single cell assay data. So something like flow cytometry or CyTOF data, where you really have a smaller number of features, but with thousands or millions of cells. And then the other reason I think Bioconductor is a natural home for single cell software development is that Bioconductor has a lot of very popular software packages for the analysis of bulk high throughput gene expression. So EdgeR, DESeq, VUM, and so forth. So we have seen a lot of growth in the last few years in terms of single cell software contributions and downloads. So on the left, we have the number of distinct IPs that are downloading the software developed by Bioconductor. And then on the right, it's the number of packages, and the colors represent the different types of assays. So in red, you can see that single cell is really taking off. And so we expect a lot of growth from here. And so what I just wanted to highlight at the beginning of this next section is that there have really been some profound changes in how users access, store, and analyze single cell data in Bioconductor, and there are really, I would say, 5 major contributions. One is memory efficient data access, import, and representation. So some of this you've already seen this morning. So things like RHDF5 from Mike, delayed array operations, standard parallelization operations, beachmat, you'll hear later. 2 are these common data containers for storing single cell assays. So namely the single cell experiment or the Loom experiments. These standard data containers really allow and enable interoperability, not only between the Bioconductor packages that have been developed for the analysis of single cell data, but also allows for interoperability, say, between Seurat, a non-Bioconductor package, and Bioconductor packages. And then there's also conversion functions that are going between the Python containers and single cell experiments. So it really enables interoperability. And then there also enables workflows. So you can go from like just a raw single cell experiment container to something that's got the reduced dimensions for either PCA or TSNE stored in it. And so it's very easy to do quick data visualization checks with this. 3, the third thing is that Bioconductor has really developed fast and I would say robust methods to pre-process and normalize single cell data. So this is one example that I recently created from a 10x PBMC data that Davide, Rousseau, Casper Hansen, and I contributed to Bioconductor. There were two data sets. One were PBMCs with 3,000 cells and another data set with PBMCs with 4,000 cells. And I just wanted to demonstrate how you can use the mutual nearest neighbors package to remove the batch effect and normalize the data. And then you can use the batch corrected data downstream for downstream analyses. So it's very easy to use these fast and robust methods. Four, I would say that Bioconductor has done a really good job of doing.
Final minute.
Yeah, okay. Interactive data visualization. So there's a package called ICY that I think is just amazing. It's used by Sarah Teichmann's lab. And then there are really state-of-the-art algorithms for downstream single-cell analysis. So ranging from dimensionality reduction, clustering, differential expression, trajectory analysis, gene set analysis, gene network analysis, ontology, and annotation. It's really state-of-the-art. And then we are working on a set of single-cell workflows to demonstrate a lot of this outside, in addition to the vignettes that have already been written. This will be used or be submitted along with the paper that we're writing to show people how you can orchestrate single-cell analysis in Bioconductor. And that's it.
Thanks, Stephanie. We'll move to the next presentation from Aaron Lund